Have you ever wondered what makes you unique? From the color of your eyes to the shape of your nose and even your talents. Why are you different from everyone else, even your own siblings? The answer lies deep within your cells in an incredible process called meiosis. It's through meiosis that our bodies create reproductive cells that make each of us genetically distinct. But what's truly fascinating is how this cellular dance gives rise to endless variation, ensuring that no two people, except identical twins, are exactly alike. Imagine a highly choreographed routine where chromosomes pair up, swap pieces of DNA, and split apart, not just once, but twice. This precise sequence results in the formation of eggs and sperm, each with half the genetic material needed to create life. In today's video, we will begin by exploring what meiosis is, followed by a detailed look at the phases of meiosis. Next, we will discuss where meiosis takes place, and then cover errors that can arise from meiosis, such as Down syndrome and Klinefelter syndrome. Finally, we will learn a mnemonic to help remember the phases of meiosis. Let's begin the definition of meiosis. What is meiosis? Meiosis is a specialized type of cell division that results in the production of four non-identical daughter cells and reduces the chromosome number by half in each daughter cell. These cells are known as gametes, sperm in males and eggs in females. Stages of meiosis. Meiosis is broken down into two main stages, meiosis I and meiosis II. Each of these stages has its own series of phases. Before diving into each stage individually, it's important to first understand the stages of meiosis using a chart so that you don't get confused while watching this video. This chart represents meiosis, which consists of two phases, meiosis I on the left side and meiosis II on the right side. Meiosis I has four stages, Prophase I, Metaphase I, Anaphase I, and Telophase I. Similarly, meiosis II also consists of four stages, Prophase II, Metaphase II, Anaphase II, Telophase II, resulting in a total of 8 stages in meiosis. We will explore each of these stages with animations to understand what happens in each phase. Stages of meiosis. 1. Meiosis I is the first round of division, and it focuses on reducing the chromosome number and creating genetic diversity. We are going to start with Prophase I. You can call it mixing things up. Prophase I is arguably the most crucial phase of meiosis, where several important events occur. Chromosomes condense, chromosomes become visible under a microscope, and homologous chromosomes, pairs with the same genes but different variants, come together in a process called synapsis. Crossing over takes place. This is a key feature of meiosis. Do you know what crossing over means? Homologous chromosomes exchange segments of DNA, creating new combinations of genes. This process, known as crossing over, this is the reason for genetic variation. Picture it like two friends swapping parts of their wardrobes. Each ends up with a unique new look. Nuclear envelope breakdown. The nuclear membrane dissolves, making way for the chromosomes to move around and spindle fibers form. Now, we move into another phase, metaphase I. You can call it lining up with a twist. During metaphase I, the homologous pairs line up along the metaphase plate in the middle of the cell. This is different from mitosis, where individual chromosomes align. The arrangement is random, which means that the way maternal and paternal chromosomes line up can vary. This randomness is called independent assortment, which adds to genetic diversity. Here comes third phase of meiosis I. We can call it pulling apart. In anaphase I, the spindle fibers pull, you can call it tear apart, moving them to opposite ends of the cell. Importantly, sister chromatids, which are the duplicated copies of each chromosome, remain together at this stage. This helps ensure that each new cell will have a mix of maternal and paternal chromosomes. Now, we're moving to the last phase of meiosis I, which is telophase I and cytokinesis. You can call it dividing the cell. In telophase I, the separated chromosomes reach opposite ends of the cell, and the nuclear membrane may start to reform. Cytokinesis then divides the cytoplasm, resulting in two haploid cells, each with half the original chromosome number. These cells still have sister chromatids attached. Now, we are moving to the stages of meiosis II. Meiosis II is similar to mitosis and is all about separating the sister chromatids. I have created a detailed lecture on mitosis. You can watch this lecture in my channel, or link can be found in the description. Let's come back to the phases of meiosis II. Here is the first phase of meiosis II, which is Prophase II. You can call it getting ready again. In Prophase II, the chromosomes, still consisting of sister chromatids, start to condense again. The nuclear envelope breaks down if it has been reformed, and new spindle fibers begin to form. Now, we come to the second phase of meiosis II, which is metaphase II. You can call it lining up for the final split. During metaphase II, the chromosomes align along the metaphase plate, much like they do in mitosis. This stage sets up each chromatid for separation, with spindle fibers attached at the centromeres. Here is the third phase of meiosis II, which is NFS 2. You can call it chromatids finally split. In NFS 2, the spindle fibers shorten, pulling the sister chromatids apart and moving them to opposite ends of the cell. Each chromatid is now an individual chromosome, ensuring that each new cell will have just one copy of each chromosome. Here is the last phase of meiosis II, which is telophase II and cytokinesis. You can call it the final product. Finally, in telophase II, the nuclear membrane reforms around each set of chromosomes. Cytokinesis divides each cell into two, resulting in four haploid daughter cells, each genetically unique and with half the number of chromosomes. Now you might be thinking, what is the end result of meiosis? The end product of meiosis is four genetically unique haploid cells from one cell. In males, these become sperm cells, while in females, one becomes an egg cell, with the other three being smaller polar bodies, which are usually not used in reproductions. Do you know where and when does meiosis occur? Meiosis takes place in the gonads, the testes in males, and the ovaries in females. This process happens during the formation of sperm cells, which is known as spermatogenesis, or during the formation of egg cells, which is known as oogenesis. The timing differs between males and females. In males, meiosis starts at puberty and continues throughout life, while in females, meiosis starts before birth and continues in cycles from puberty onward. Now, let me tell you what is the purpose of meiosis. Meiosis has two main purposes: reduction of chromosome number. It reduces the chromosome count by half from 46 to 23 in humans. This ensures that when a sperm cell and an egg cell combine during fertilization, the resulting zygote has a full set of 46 chromosomes. Genetic variation. Meiosis creates genetically unique cells. This variation is the reason why siblings can look so different from one another, even with the same parents. How mistakes in meiosis can affect us. Sometimes mistakes occur during meiosis. When chromosomes do not separate properly, it leads to conditions like number 1 Down syndrome. This occurs when an extra copy of chromosome 21 is present due to a failure in chromosome separation. Number 2 Klinefelter syndrome. This is the result of an extra X chromosome in males. These errors are rare, but they highlight the importance of the precise control involved in meiosis. Tips for remembering the phases of meiosis. Here's a mnemonic to help you remember the stages of meiosis. Prophase, metaphase, anaphase, telophase. Repeat for 1 and 2. An easy phrase could be, "Pass me another taco." Then remember there are 2 rounds. Question of the day. In which phase does crossing over occur? If you are a genius, you can answer in the comments below.